Thank you, everybody, for joining. And I'm sure we'll have a few more join as the time elapses. But we'll go ahead and get started to kind of be punctual with time. Just as a small announcement, this webinar is also being broadcast again tomorrow morning. So about what are we doing? We're doing about 14 hours from now. So we'll probably get some nice attendance there as well. So anyways, my name is Derek Novak. I'm going to be hosting and introducing our guest speaker today, Laura Otter. She's been working with us and doing a photo-induced force microscopy in the geosciences. And so our webinar today is going to be looking at those applications. I will start out with a short introduction, somewhere on the order of about 15 minutes or so, highlighting what PIFM is, what it is we're trying to accomplish with the technology, and what applications we've done, particularly in the geosciences, but also, try to give an idea of what PIFM is capable of. I added in a few other little applications that highlight its extreme resolution capabilities. After that, we'll switch over. Laura will present to us work that she's recently done, partly with our help and in collaboration. We've done collectively here at Molecular Vista, and presenting a nice application that she's done in the geosciences as well. So I'll hold that until we get to her section. But let's go ahead and dive in and get started. So effectively, what we're trying to do with photo-induced force is be able to measure chemical information down at the nanometer scale. So we want to be able to take a sample and deconstruct it down at the tens of nanometer style resolution. So we want to circumvent kind of where traditional FTIR gets stuck, where we can only really come down into tens of microns in terms of resolution. We want to go 1,000-fold lower than that and get into the tens of nanometers resolution. Now we do this by employing an AFM cantilever. And so if you can kind of think about the way that an FTIR system might work, is you would have light coming in radiant on the sample surface. We'd have a spot size associated with that radiation, which would define your resolution. But if we put this probe in place, we think it can then spatially resolve within that focal spot at a much smaller resolution, a resolution that's really limited by the probe geometry itself. And that's the kind of cartoon you're seeing here. So to try to keep it somewhat simple. But we go a step further and remove traditional difficulties that you would have in near-field imaging or imaging where you're trying to probe within a focal spot. A lot of times what you're going to fight against is scattered light that's scattering off the sample, light that's scattering off the tip, and that's confusing. You don't know if it's coming from that near-field region that you're interested in. So what we do is we look at a force-based measurement. We try to feel the absorption in the sample right underneath the tip. And then by doing so, we can now be limited to just the resolution of the end of the tip. And so we're going to do that using kind of a multimodal schema in AFM. And so you can envision our AFM cantilever having multiple mechanical modes, but we'll start with the simple first mechanical mode and a second mechanical mode. Now what we can do is we can use the second mechanical mode to investigate the topographic information of the sample, and that's what you're seeing in the gray image. On the first mechanical mode, we can start to detect the absorption event in the sample because that absorption event is going to pull on the AFM tip itself. So as that sample absorbs, we feel this attractive force, it pulls on the tip. And then we can start to generate images that correlate to the absorption at different wavelengths of light or wave numbers of light, depending on if you have more of a chemistry background or more of a physics background. So here we're looking at PMMA absorbing at 1733. That was part of this composite of materials in this topography image you see here. We can see polystyrene and where it's located. And then we can also see aluminum oxide and where that's located. So this particular sample was a composite of PMMA, polystyrene, and aluminum oxide. And just by changing the wavelength of light, changing the absorption response in the sample, we can then go in and probe and tell you where those chemicals are physically located. How can we take this and now start to use it in a space where most analytical chemists are going to be quite familiar? Well, nicely, and is that there's a great correlation with FTIR-based results and our PIFM-IR results. And so what we're doing here is we're taking that same laser source that would be tunable from, let's say, 800 wave numbers to 1800 wave numbers. And as we tune the laser source, we observe with the tip what the optical response of the sample is. And so we go through and we observe this response, and we can see when we take that, go and look at it with an FTIR machine, that the correlation is very, very good. And we've now found over the last few years of working in this space that we can easily take spectra that we acquire and we can send that to know-it-all or a Wiley type FTIR library, and then use that information to actually ID chemicals at the nanoscale, particularly chemicals that were unknown to us. Anyway, so you see a nice correlation with the polyamide, nylon, PES, and in this case, we looked at a polyethylene oxide material as well. So let's kind of put this conceptually into a picture to show why it's important to be able to get down to the nanoscale. Imagine this topographic image here is something you want to image, and you want to look at, okay, what materials are here. So you put this into an FTIR machine, and really your spot size is going to be about this red circle you have here, just estimating. And you get this nice red response, this FTIR response from the sample. But that is just a bulk information. You don't know really what constitutes this ensemble of material. It's just telling you that this is the ensemble. But if we start to go in and take, let's say, point spectra at different locations within this ensemble, you can see that the PIFM is able to start to deconvolute or pull out unique spectra associated with different materials within that composite. Right, so you'll see at location 1, we're seeing something that looks quite similar to, let's say, like a silicon oxide, maybe a silica particle, and then we're seeing more of an organic type nature in the light blue and the dark blue locations. So just to give you a picture of what it is we're trying to do. Now, how sensitive can we get? So this is some very recent work that we've done with researchers at LBL, Lawrence Berkeley National Labs. And what they were trying to do was create a monolayer of these peptoid molecules. Now, these are quite big molecules. We're talking about roughly 3,000 molecular weight style molecules. So they're quite big macromolecules. And it was quite interesting because we start looking at their topography and we're looking at the PIFM results. We know there's going to be an absorption event around 1633, an amide, kind of near an amide type protein measurement. And what we were kind of dumbfounded by is we're like, basically weren't seeing much at all. We're saying, well, we don't really see your molecules. It's supposed to be a uniform sheet based on the predictions from the group. But we started to identify small little islands that were lighting up in the PIFM. And if you look closely, we're looking at, let's say, this green arrow here, this red arrow here, and here we can tell you the heights because of the PIFM measurement. So we're really talking about something, particularly the red arrow, where we're about 1.2 nanometers tall, and the width's about 16 nanometers. That's going to be a little bit of tip convolution from the AFM, most likely. So the molecule could be slightly smaller than that, for sure. And we're seeing a definitive result spectrally at those points. So we're parking at those locations, we're taking a spectrum, and you're seeing, a characteristic response that we would expect from those peptoids. Now, so what it was telling us was this is not uniformly coated. During the process, a lot of them were removed and just a few particles were remaining. But now let's take a minute and look at what the spectra were actually telling us. So you can see that the green dot has definitely a stronger spectral response and the red a little bit smaller. Well, you would expect the concentrations are smaller. The peak ratios tend to try to stay roughly the same. But we're expecting a strong response from these peptoids in this kind of FTIR region that we're seeing highlighted in the blue bands. And we are seeing that correlation. Now, you could ask, why are you seeing these other peaks here? So we have this peak around 1260. We have a peak around 1380. And then we have another peak down here in the 810 kind of region. So these we're finding are contaminants. So we have some siloxane-based contaminant, we have some plasticizer-based contaminant, and apparently these were part of the process in making the samples. So not only were we seeing the molecules themselves that they're interested in, we were also seeing artifacts from the sample creation, these pre-processing events that we might have been using siloxanes or other plasticizers. So really powerful and eye-opening for that research group. So just to kind of expand on what PIFM can do, I'm going to introduce just briefly a hyper-based measurement. And I do this because the next application I show does involve using a hyper. But you can envision now that we've taken these point spectra on a sample, and what would prevent us from taking a full array of spectra that allow us to create a three-dimensional grid or four-dimensional grid of where we now have each individual pixel in our AFM topography represented with a spectrum to go with it. And so we're able to do that. Certain laser systems, we can do that easier than others, but in our kind of molecular fingerprint region, we can actually scan the lasers quite fast. And in that region, we can then go through and take a full spectrum at each pixel. So what does that kind of result look like? So this is a very early result we had done in the company with these hyperspectral videos. And let me start this video here as I continue to chat about it. So what's going to happen in the video you see here is it's going to be slowly changing through wave numbers and giving you an image at each wave number. So we have this hyperspectral data set. And as we tune through these various wave numbers, you can see the image change. You can see what parts of the image are absorbing at those wave numbers. And I still, after the years that we did this years ago, and the thing that I still like about this result is that you will see things in the hyperspectral video that you don't see in the topography and you don't see in the phase. And so really without that ability to analyze the sample with all these individual spectra, you might be missing something. You might not see everything there is to see. And so we kind of go through these and you can kind of see where these things highlight. And it's just a really powerful tool to learn something about a sample, particularly if that sample does not have a lot of prior knowledge associated with it. So I take that same data set, I just pull out 5 images just to show, we've got this hyperspectral cube of information. We can then go in and pull out slices of wave number images. And again, all trying to do this at that sub 10 nanometer resolution. Okay, so we go through that hyper idea. Where was one place in the geosciences where this was really helpful was looking at shale. And so this was, again, kind of an early study we did. And the interest was in the mineralogy and, of course, the thermal maturity of the shale. And let me kind of go to how we attack that problem. So we worked with some researchers that effectively were able to identify what wave numbers were associated with, let's say, quartz, what wave numbers would have been associated with clays, carbonates, organics, and quite a few different wave numbers that would, of course, be associated with organics. And so here's kind of an initial approach at it. We have this topographic image, we have this hyperspectral data set, and here we just pull out 10 individual spectra out of that hyper set, and those are plotted in C. And you can kind of see how you're getting different peaks at different locations. We can go through, we can image or pull out of that hyper cube, a wave number image at 1100 and one at 1265. So we can start to use this as ways to determine where are we seeing organics and inorganics in that shale rock. So we did this on 4 different samples, and we built a composite image of purely inorganics. So all the images across the top are just highlighting where inorganics were visualized in that data set. And while it's not highlighted here, just to give you an idea, these images are on the order of 7 by 7 microns, right? So we're just at the limit of the whole image being, an FTIR spot. But instead, we're going into that that region, that diffraction limited region, and working our way into the sample itself and looking at that nanoscale information. So again, 4 different samples, 4 different kind of distributions of inorganics, did the same thing on the organic absorbing wave numbers, and you can kind of see how those are distributed, right? So this was just kind of one of these survey studies. What could we pull out of the systems? And then the idea would be, can we start to use this information to understand thermal maturity, mineralogy, and make better, kind of more informed ideas on how one might extract shale from different locations around the world. Okay, so then one other little thing I would highlight here is meteorite imaging. So we did a kind of quick study with a group in France. They sent us some simple meteorite samples that they had prepared. Now, of course, the idea here was, could we see organics within the meteorites themselves? Of course, that would have a pretty nice impact scientifically. I'm not going to be here to make that claim today, only because, humans touch these things. So you always wonder where the DNA comes from, but or where the organics are coming from. But to give you an idea of really the power of PIFM and the ability to get high resolution spectral information, this is just a great example. So we start out with a fairly large image. We're talking about maybe a 5-6 micron image. We zoom into this red box region, and we take a series of point spectra along that line you see here. Now that data is presented here in all the spectral lines you see, going from 0 down here to 40, which is going to correlate going from the upper part of the image down to the 40. And you can see how chemically we see a variance based on where we're located. Well, so certain peaks will be quite strong. They'll decay for a while. They might return, kind of depending on where we're at on the sample itself. So we're looking at not just the distribution of where these 3 materials that we've highlighted are, that you see in this combined PIFM image, the silicon oxide, the phyllosilicates, and then this hopefully some kind of RNA, DNA thought, or at least proteins. And you can see then that is not evenly distributed. It's not homogeneous. And you're seeing that same response here spectrally, that certain regions within that smaller box will have stronger responses at 897, as shown here, or at 1035, or at 1095. And those we've assigned, accordingly to the silicon oxides, the phyllosilicates, and so on. So again, just to kind of really showcase the power of that kind of sub 10 nanometer or 10 nanometer spatial resolution and spectral resolution. Okay, and so to kind of wrap up my side of the story for today, before we switch over to Laura, recently we just started also looking at nanoplastics. And so since this has a large environmental impact, I thought this would be good to showcase also in a kind of a geoscience talk as well. And so this is more of a test case. This is where we're working with some colleagues at McGill University in Canada, and they said, okay, we're going to make you a mixture. So here's a mixture of PMMA, PTFE, polystyrene, gold, and we're going to let you go ahead and image those, and let's see if you can identify where those different materials are. And so you can see very nicely, if we take a point spectra on top of this kind of large nodule here, we get a very nice Teflon spectrum, or PTFE. If we highlight one of these smaller bead regions over here, we see a polystyrene based spectrum. And then, of course, if we highlight the smaller ones, we're seeing PMMA. And you can see that again if we take fixed line images at each, at a wave number that is unique to one of those 3 materials. Now, the gold's a little different. Gold's not going to have a nice IR response in these wave number ranges. So you're going to get a little bit of contrast for gold at all wave numbers because it's going to be pretty flat. So here we're just highlighting off resonance. So at 1800, where nobody has a resonance absorption peak, where we're seeing potentially gold. Now to visualize this a little bit better, we take that same image you saw on the previous slide. We've highlighted now in blue where the PMMA is located, where the polystyrene is located, and where we're seeing where we expect gold or an PTFE. And so really, in some ways, this is a nice kind of review in that, these are PIFM is really great for nanoplastics and really any IR responsive material, in the sense that it's able to kind of characterize both size and chemical identification for these particles. We also did that on that peptoid molecules as well. We're easily able to identify organic and inorganics. Now that's true kind of across all the applications that we do. And its ability to kind of look through kind of thin layers of biological contaminants. So, a lot of times what you'll find on samples is that they might have some kind of contamination layer on top. Maybe it's from the preparation, the storage, but we do have the ability to kind of peer through that. We're surface sensitive for sure, but we can see a little bit through those surfaces and then identify without fighting those contaminants. Of course, since we're doing all this in the IR, we don't have to worry about any kind of fluorescence issues. Something you might deal with a Raman if you're trying to do similar studies. Again, we're non-contact, non-damaging measurement. We don't physically touch the surface in a way that's going to damage the surface. And in general, it's like an AFM in terms of its requirements for sample preparation. So for fairly generous sample preparation in that sense. No special coatings, really no special pre-processing that's required. If it works in an AFM, it's going to work in the PIFM as well. Okay, so without much ado, that was my kind of intro into PIFM. Hopefully I didn't take too much time there. But really, the point of what we'd like to do today is get this this talk from our guest speaker, Laura Otter, which we are very, very happy that she's able to join us today. This is great. She is in Australia, so we're trying to navigate the world today. But just a little introduction to Laura. We've been working with her for a few years now. It's really been a nice treat to work with her group and herself. She's a geochemist by training. She started out her career and her education at the Johannes Gutenberg University in Mainz in Germany. And then I think from that point, she got to do work then at Max Planck Institute, also in Mainz. And then it looks like really got into, looking at biomineralization and into the kind of marine mollusks and looking at cultural pearls. I think that's a little bit of the results we're going to see today and the talk that she'll give today. She did a lot of this work when she moved to Australia with her advisor and was able to finish her PhD there. Laura, if I'm doing anything wrong here, feel free to correct me when you start. But so now, she spent a lot of time at Macquarie. That's where we met and started interacting and collaborating. And now she is at the Australian National University and will continue doing PIFM studies as well in the laboratory there. So with that, let's try to switch over to Laura. And let me stop sharing my screen. I think I'm off, so go ahead and feel free to bring yours up.
Can you see my screen, Derek?
Absolutely, and I can hear you well. So I'm going to mute myself, but I'll unmute if something comes up.
Okay, fantastic. Thanks everyone for joining, and thanks for the nice introduction, Derek. That was all correct. So I'm going to talk about how to apply PIFM into to biominerals. And without further ado, I'll just jump into this and give a bit of a recap of the PIFM development. So it was the first publication looking at PIFM was published in 2010, just on the beginning of this chart here. And then we saw more method development popping up since 2014, and that's still ongoing, and first applications coming out since 2014. And these have been, in the majority of cases, in the areas of physics, chemistry, and biological sciences. And the first geochemical application that Derek mentioned too was published here in 2018 by this group, looking at thermal maturity of organic phases in unconventional reservoir rocks. So, and then sort of the next step into the geochemical area was done perhaps with our review paper that you can see here. And that was just published earlier this year, so just off the chart here. Okay, and in this review paper, we looked at zonations in zircon, fluid speciations in high pressure experiments, and biominerals. And this is the part I'm going to show here. So, but now talking about PFM and how it complements traditional geochemical methods. We have a bit of phase ID perhaps that we use in the geosciences, and these are here circled with these dark circles. These are XRD, FTIR, and Raman, but they're all kind of in this area of low resolution. And there is currently no real high resolution phase ID technique available to cover this sort of part of the plot. And this has been just a gap between the phase ID on one hand on the low-res plot and the high-res area where we just have element analytical methods, circled here, for example, the atom probe tomography, TEM, nanosims, and so on. So there's this gap. And now PFM is a method that can sort of fill this gap and can carry like the phase ID techniques down into the high-resolution area. So it sort of extends traditional phase ID, and we can do everything that we did beforehand in traditional phase ID, but just at much better resolutions, lateral resolutions of about 5 nanometers, and still at the same spectral resolution of about 1 wave number. We have a low depth penetration, which also helps in getting really high resolution imaging of different phases or features of phases, such as zonations. And as Derek said, PFM is non-destructive, so there's no sample consumption or beam damage or coating or any kind of modification of the surface necessary to do this work. So because PFM uses so low energy laser outputs. And the way I think this would ideally integrate into like existing geochemical workflows is like by continuing to do optical microscopy pre-characterization techniques that we have all been using all the time, then doing PFM because it doesn't change the sample at all, and then carry out any kinds of methods that need a bit of change to the sample surface, such as coating or destructive analysis, like APT, for example. So, and then I'll get starting on the applications to biominerals. And biominerals are just any kinds of minerals that are produced by living organisms. And this could include like bones, shells, teeth, and anything. But really prominent examples of biominerals that have a geochemical significance are the shells of marine calcifiers, like bivalve shells, brachiopod shells, corals, plankton, and so on. And here in this talk, I'll just focus on the shells of bivalves, such as mussels and clams, cockles, oysters, and so on. And these bivalve shells come in 2 different types. We call these architectures. And this means that they have different 3D hierarchical arrangements of the calcium carbonate phase on one hand, and the organic phase on the other. And here we will focus on the architecture called naika. And in the images here, you can see on the top some non-nacreous shells that have this lag cluster inside. And then down below here are the nacreous shells, and they have this shiny interior on the shells, or we see a naika also on the outside of pearls. So, but why am I interested to study naika at all? So this is diverse in terms of motivation. There are aspects of sclerochronology, material science, and gemology. So, and I'm interested to study shells to learn about how we can better understand the applications of paleoclimate records. Shells grow these little increments that we can measure for trace elements and isotope signatures to reconstruct past oceanic conditions. And this is one part of the motivation. The other one sort of falls into the realm of material sciences because shells are so mechanically strong and crack resistant, and we can't actually make materials so light and strong with just simple phases like calcium carbonate and a bit of organics ourselves at low pressure and temperature conditions like the bivalves. So it's interesting to learn how shells grow and then sort of try to adapt this into our material sciences to make better materials. And the other aspect is, of course, because naika makes pearls or is part of pearls. This a better understanding of naika means also a better understanding of the pearl culturing process and the different gemological properties of pearls. And we can see some nacreous shells and pearls here. And let's have a look at how this architecture looks. So this consists of polygonal tablets made of calcium carbonate, and that are sort of situated between layers, the stack material here, of organic phases. And in a cross section, this looks like a brick wall, which is why this is also called the brick and mortar architecture. So, but now, why did I wanted to try using PFM to study biominerals? This has mainly two reasons. Nacre is a very beam-sensitive insulator, and this causes problems with E-beam techniques and lasers because they can induce dehydration and phase transformation that lead to structural alteration, which we actually want to avoid. So, and PFM uses these low laser energies of less than 1 milliwatts, and analyses are also performed at ambient pressure, so there's no vacuum involved, which would also change phases, perhaps. So this is ideally suited to look at hydrated, sensitive samples like nacre and other biominerals. And the other aspect is because of its architecture being a nanocomposite, meaning that it consists of organic and inorganic phases that are arranged in a 3D hierarchy, that we can look at both phases with this technique because they have this very good resolving power. So we can go down to 2 nanometers depth penetration or have a very good spatial resolution of about 5 nanometers. And this is great to resolve the structures that are usually not observed in many other methods. So, and this brings me to showing you some results. This is a pearl that we just cross-sectioned and polished. This is mounted in epoxy, as you can see down here. And when we look at this with an optical microscope, this is how this looks. So we have a bead on one side. This is what is put into the bivalve to grow a pearl. And the bivalve then grows this layer of nacre on top of the bead. And this is now just sandwiched in between the bead and the epoxy because this whole sample was just mounted into epoxy resin. And this was the first image we got of the PFM. This is just the AFM topography to give you a sort of an introduction to our results. And this is sort of the lowest resolution map we did. And you can see that the light colors represent the calcium carbonate, and they're a bit elevated here. And then darker recessed are the organic phases. And this is the organic membranes that I talked about earlier. So this already, although it's like the lowest resolution we could do, really resolves the structure very nicely. So, and now showing you also the topography a bit more zoomed in combination with PFM maps. We see in this PFM map here in the middle, on the left-hand side, we see the calcium carbonate peak that was mapped. And this really lights up in between these organic membranes and really resolves the calcium carbonate phase nicely. And on the other hand, the organics show up here as this brighter membrane areas as we map the Ahmed peak that is part of the proteinaceous material. So, and how does this compare with what we know about nacre? In this SEM image here, you can see also the calcium carbonate tablets, that they're enveloped by these sticky-looking organic phases. And while this can't tell us much about the phase ID, this is just an E-beam image. We see here with PFM, we can reproduce this on the polished cross-section and pick up all the different phases in the nacre. So zooming in further, this is now a field of view of 1 micron by 1 micron. And here's the AFM topography again, and we can see 2 nacre tablets on either side of this darker organic membrane. And this in the middle is again this PFM map of the calcium carbonate phase, and we can see that the tablet lights up very nicely, whereas there's no calcium carbonate in this membrane area. But there are also some speckles in the calcium carbonate phase that I'll talk about in a second. They were also visible on the topography as these darker recessed little speckles here. And then we had another PFM map of the Ahmed peak, and we can see the membrane area actually being rich in this proteinaceous material. And this is also showing up as these little speckles that we observed previously. So all these little spots showing up here are also proteinaceous, and therefore they don't have calcium carbonate, and that's why they're dark in here. So another way we can look at PFM data is just to stack all the different PFM maps together to form a composite map. And this is what we see here. And the blue color shows the calcium carbonate peak that was mapped, and that's mostly showing up in between the membrane in these 2 different tablet areas. And then we had the Ahmed peak that shows up mainly in the area of the organic membrane and also in these little speckles of the calcium carbonate. And we added a 3rd peak here showing water, which is sort of making up the rims of the nacre tablets that we see here, so either side of the membrane. And this is a very nice way, I think, these PFM composite maps to get a really detailed understanding of the phase composition of the sample. So, but what we can also do is collect spot measurements. So here we went in and took a spot measurement here from inside the membrane and one just outside the membrane in the calcium carbonate part. And this is what we get. We get a very nice FTIR looking spectra of the organic phases. This is the amide peak region here. And this peak, this long one here, is like from the calcium carbonate phase. And when we compare this to traditional FTIR, which I'm showing on this side here, just obtained from another pearl, and this is just popped upside down because this was obtained in reflectance mode, we can see that FTIR is actually showing less of the peaks. They're more convoluted looking, but otherwise it shows the same peaks. So PFM actually corresponds very excellently with the FTIR data, and just peaks are looking a bit more narrow and therefore less convoluted, which is nice. So, and another way to look at PFM data, which I found was very exciting, is that we can actually stack the 3D topography together on top of the PFM composite map. So what we get then is like a 4D data set showing the chemical information in sort of a 3D setting. So we can have a perfect reproduction of the sample surface. And what we see here are these 3 peaks again, and we can get a better understanding of the organic inclusions this way. So I've just circled a few that were also visible in the topography map here as these little darker dots because they're recessed. And these were like opened during the polishing process, but there are also some marked by these triangles that don't have the corresponding peak in the topography. And we think these are the ones that are not open during polishing, and they give us actually a stronger amide signal. So these are organic inclusions with the original organic content very well resolved. So, but proteinaceous inclusions in naika, how does this make sense? This beautifully integrates with other data that we produce. So this is a TEM image, and with the lighter color showing the organic membrane and also the lighter color showing the organic inclusions as these little speckles in the darker matrix of the calcium carbonate map. And this fits very nicely with previous studies showing that these inclusions are visible in TEM images. And also a previous study that showed that inclusions are rich in carbon. This was a TM EELS work performed. And also our previous atom probe tomography work could show that these inclusions consist of carbon, nitrogen, and hydrogen, which again just underlines the amide nature of this proteinaceous material. And so actually PFM data is very well complementing everything we did with these element analytical methods. So, and the last question, I guess that we need to ask is, why does naika have inclusions at all? And we found an answer published here in our Nature Communications paper, where we just stuck a bit of naika from the Spinanobolus shell into a TEM and doing nano indentation by viewing this live in the TEM. And what we could see was that during compression of 40 micronewtons, we saw that tablets, the calcium carbonate tablets, interlock across the organic membrane. That's a bit squashed inside the tablets then, and that these little inclusions rotate and deform under pressure. And after taking the load away, everything just jumps back and looks perfectly like before the experiment. So inclusions just complement naika's mechanical properties and make it actually very resilient and crack resistant. And yeah, this brings me to the end of the talk, and I thank you for your kind attention, and Molecular Vista and the Geochemical Society for having me talk here today.
Well, thank you, Laura. That was very nice. Actually, I have 2 questions right away, if you don't mind.
Okay.
First off, actually, before we get started there, this is a new platform for us to work in. So for those in the audience, if you do have a question, you should have, I'm going to bring my screen over a little bit and share my screen for just a moment so I can show you. If you look in the webinar itself, there should be a little Q&A option that you can click on, and that should allow you to actually ask questions of the speakers via text. We don't have the audio turned on at this point, as we kind of learn about this platform. But So let me go ahead and ask my two questions, and then if anybody in the audience has questions, please do write us, and we'll answer those as well. But the first one is on the polished cross-sectioning that was done. Was there any special preparation, or was it just really kind of mechanically polished?
Yeah, so what we did was just cut the pearl with a low-speed precision sectioning saw, and then just polish it with diamond pastes down to 1 micron. And then what I usually do is like adding a step of colloidal silica that's 0.05 micron, I think. And then this gives me like a very nice finish to do all kinds of other imaging at high resolution.
Yeah. Well, if you look at actually, if you don't mind, let's go back to your presentation.
Okay.
And if you look at the topographic heights, I mean, we're talking just tens of nanometers on those polished cross sections.
Which one would you like to see? This one?
Actually, any of these are quite fine. I mean, even the image you have here on the left, really shows just 10 nanometers. So the polishing seems to work quite well. And I just wanted to highlight that for those in the audience that, again, this kind of highlights that ability to not require anything special in terms of sample preparation. Now, if there's something where you're going to be worried about material spreading across the sample as you're polishing it, then you may go to ion beam or something like this to kind of clean it up. But we've noticed in the geosciences, a lot of just polished samples work quite well. The other question I had, just watching your talk, so slide 12, if you don't mind, the two spectra that you show.
Here we go.
Now, you had made a comment about how the FTIR is a bit smoothed. Now, are these materials, are they crystalline in any way that would cause their absorptions to actually be narrower than what FTIR might predict?
So the calcium carbonate is crystalline aragonite. I'm not sure how this. Yeah, good question actually. Yeah, not too sure about the physics behind this really.
That's okay. We have found in like quartz per se that if it's a crystalline quartz, particularly based on the kind of crystal zone orientations, depending on how that crystal is formed, we will get different spectroscopic responses. But if you were to look at it as an ensemble, then you would get this kind of smoothed FTIR response.
Yeah, so with naika, the c-axis is always aligned the same way. So this is basically looking down the c-axis, but the A and B axis would vary with each little platelet. And because FTIR has like this huge spot size, so to speak, I guess there's a bit of averaging out between two of the crystallographic axes. Yeah, I guess that's contributing to the smoothing.
Yeah, okay, that makes sense. So there could be something there to learn then, and how how that structure looks down at the nanoscale opposed to the macro scale. Interesting. Yeah. I mean, I know that we did a little bit of co-authoring on this paper, but yeah, there's definitely some details in there that are quite intriguing. So very cool. Well, I don't see any questions coming in. I'm hoping that's not because we are doing this wrong. It could be quite possible we don't understand the platform well enough.
There is a question.
Do you see one? Oh, very good. Where is this? Thank you for chiming in. Where do you see it, Song? I'm sorry.
In the question and answer box that you mentioned.
Oh, excellent. Yes. Okay, it works. Dorit, thank you for helping. Okay, so Dorit's asking, it's kind of more of a general question for the both of us. So how careful do you have to monitor organic contamination of the sample? And then go a little bit farther, would the contamination risk be different for composite of organic, inorganic material than purely inorganic samples? Interesting question. So maybe I take a stab at it a certain direction, and then I'll let Laura maybe try a different direction. from a PIFM perspective, we have a couple different ways to guarantee that the measurement probe is not contaminated. So we can take special care. One is in how we do the preparation of our cantilevers, the storage of our cantilevers, and then also we can kind of pre-measure them on a clean material. So we can take a like a graphite or something like this. We can guarantee that it's freshly cleaved. It's got a clean surface where no contamination could have landed yet. I guess you could do a similar thing with mica if you were to peel mica. And so we can start out knowing that there's no contamination there. In terms of, so for monitoring, at least from the PIFM side, we can kind of guarantee that there's no organic contamination. Now, from the sample prep side, I'll have to leave that maybe a little more with Laura and how one might prepare it to make sure organic contamination doesn't become a problem.
Yeah, I guess this just needs a bit care of how to store samples and so on. I guess plastic containers are not so great because there could be some outgassing of the softness and things like that. I think we had that issue sort of in the past at some point. Yeah. But I guess like any kind of glass or aluminium would be good. Do you agree, Derek?
Yeah, I mean, certainly anything that's not going to outgas. There are certain containers you can use. Fluoroware is a good one. That's heavily used in the semiconductor industry because it does not contaminate samples. And we haven't seen a strong signature there in samples we've seen stored in those materials. Polypropylene actually also works fairly well.
Okay, hot plastic,
container. So.
We just weren't very lucky with these membrane boxes. Everything that's like softer plastic, I guess, is has to stay out.
So no, it's a good question, and it is interesting because it can, at this level, can really highly influence your measurements, and give you kind of the wrong results. We've looked at many samples from other clients, customers who've, we see siloxane. And it's just because, really in the storage or creation of the sample, it's exposed to siloxanes, which are, pretty ubiquitous in society nowadays. It's pretty easy to contaminate things with siloxanes. A plastic bag, the inside has siloxanes to keep it from sticking, so you can peel them apart. So things like this, you have to be quite concerned with at the nanoscale for sure. Good. Any other questions? Does any of the other, the moderators, Sung, if you had a question you wanted to ask of Laura, you're welcome to. Otherwise, I give everybody maybe one more minute, and if we don't have any other questions, we'll conclude.
Many times people want to know how long it takes to take a spectra or a map image.
Yeah. So typically, to get the kind of spectra that you're seeing in the webinar we've shown here, we're operating around 15 to 30 seconds to acquire a 1-point spectrum. And then to do a map, we're typically scanning on the order of 0.5 to 1 hertz scan speed. So if you're, if you're into AFM lingo, that's going to correlate to like a 3 to 5 minute imaging, to create one image. Yeah, and so if we're going to do something, let's say hyperspectral wise, we can typically do maybe a 128 by 128 pixel grid, and that's something you can do much faster. We don't need to do the 15 to 30 seconds per spectrum. Obviously, that would take quite a long time, but those we can acquire in in the hour to 2 hour time frame. So those typically can come in pretty quick. And again, it's a good diagnostic tool if you really don't know much about your sample and you want to start poking at it and seeing what it's composed of. Okay, well, let me start with a few finishing remarks, and then we can conclude for this this session. So I'm going to go ahead and share my screen one more time. And of course, I want to thank everybody for taking the time to join us today. If you missed some of the webinar and you happen to be up in 14 hours from now, you're welcome to join us again. We will be making the video available offline as well. So if you need to come back to it or there's something you want to do.
There's one more question about how to deal with changes to the composition and structure of the sample by resin and polishing powders.
Ooh. Oh, I see. Great. Yeah, so these don't update on my screen as fast as yours, so I'm glad that you're pointing these out. So how do you, yeah, right. How do you clean the sample after polishing to ensure there's no residue from polishing powders? Ultrasonic bath required.
Yes.
Is that how, that's how you would do it?
Yeah.
Okay.
It's also important to remove the little colloidal silica particles, so they stick otherwise quite well to the surface.
Okay.
So this will just be flushed under RO water or any molecule water or whatever available, and then just popped into the ultrasonic for a few seconds, really just.
I see. I see. And is that something that you did with the samples we looked at together?
Yes. Yeah.
Okay. Excellent. So there you go.
But we'll be keen to play around with this a bit. So maybe just trying to look at freshly fractured naika maybe is definitely something I want to try, and just see how everything looks without doing extensive treatments via polishing and so on, and compare results. Yeah.
Excellent.
I think for those who are possibly casting the sample into a resin, then when you microtome or ultratome such a sample, it might be prudent to do it in a cryogenic temperature, because then the resin is hardened and the chances of resin spreading over the sample is lessened. And such a spread, of course, will not come off even using ultrasonic cleaning, because it would be adhered pretty nicely. So in that case, the best thing is to do it in cryogenic temperature so that it's not spread over the sample in the first case.
Yeah, that's something, that's a good point. So we are able to cool our plates during polishing, and that hopefully helps. But I've never used a microtome, but I guess, yeah, because the whole blade going through the sample, I guess it would like just drag some organic of the resin maybe along. So that's a good point there. But so far I didn't try this for the biominerals yet, because I've heard from a lot of people that it's difficult for these composite materials, because the organic is quite hard for the microtome, whereas the organics are soft, and just this difference of hardness can introduce some damage at nanoscale. So that's why I never tried this, but it would be maybe nice to just compare a few different ways to prepare samples and see which one is like the best way to move forward.
Yeah, I see there's another question as well in another chat that's part of this group, but it's a similar question, so I think that was well covered. Excellent. Well, Sung, thank you for adding in that little extra bit with the sample prep. That's quite insightful. But just let's go ahead and wrap up since we're hitting just about the 1 hour time frame, just a few minutes away, so we can finish. But I do want to thank everybody for joining. So thank you for watching. If you have any other questions or want to know more information about PIFM, Molecular Vista, that's us who are the the experts in the photo-induced force microscopy. You can come to our website. Some of the stuff I presented early on, those examples are also on our website, and with a little bit more text there if you want to read into some details. If you're interested in just reaching out to us, you have an application that you like. It's not quite what we presented today, but it's your focus and your interest. Reach out to us. We're always interested in new applications, or we may have looked at similar applications in the past, and we can interact and see what's the best way to get more information out of your samples. And we do that in a couple of different ways. Of course, we do manufacture and sell instrumentation. We also do collaborative work scientifically with groups, especially in new application spaces. And we also do contract analysis work where we'll image samples for a fee. So these different options, but don't hesitate. If you're interested in just learning and talking to us, we would love to know more about what your interests are and what samples you're working on. So with that, I think we'll conclude. So Laura, thank you so much for joining us today.
Thank you.
I look forward to the next time we can do this together. So, all right. With that, I'll close the meeting. Thank you, everybody.